Hello, everyone. Hopefully you can hear me. I'm sure someone out there will signal me outside my office. Oh, I got a thumbs up. Good. Good afternoon, everyone. My name is Dr. David Taylor. I am the VP of Research for ALS Society of Canada, and I am here to present to you the 2019 update on the impact of an ice bucket, which was a webinar that I gave a little while back. We're going to do a more high-level version of it today. So without further ado, let's get moving. All right, I'll just depend on my computer advancing slides. Okay, as I always do, here's some references from images used. We're going to use these for educational purposes. So if anyone has any questions about the images that are used, the referencing, please contact us. I'd be happy to remove them if you would like. Now, the last time I gave an ALS Ice Bucket Challenge webinar, it took 3 hours. And that's because I want to go through and really comprehensively discuss all of the different projects that we have funded through the Ice Bucket Challenge funds. Today, I'm going to try to do, not 3 hours, don't worry, a little shorter of a journey. For anyone that's expecting maybe a general analysis of where the ALS field is at, I would suggest maybe going back to some of our other webinars that are online. For this, it's really going to be about where your investments have had impact and where they're going to continue to have impact in the future. I think that's really important for ALS Canada to be able to let everyone know that we have very wisely and very thoughtfully invested those donor dollars, which were such a tremendous gift, and that they are actually paying dividends in our understanding of ALS and our ability to eventually treat it to a more effective manner. So I'm going to take you a little trip back here to September 2013. And actually, we were in a moment in time in our ALS Canada research program where we, weren't quite sure where we were going to go next, because we had some previous partnerships with the Canadian government and other organizations that had kind of been dissolved for various reasons. And so we actually had our very first strategic planning for research, which was really great timing because it was less than 1 year later that the Ice Bucket Challenge hit. It really led to what we wanted to term at the time a translational grant program that involved these grant types, these competitions and types of grants that you see in front of you on the screen. And many of them that we had the actual funding for. One of them that was actually sort of, we don't know how we're actually going to fund this, but we're going to move forward and we're going to try to find new funding for this. But we really had about $1.5 to $2 million that we can annually invest in research based on fundraising that we had across the country for our national research program. What this really did is it set us up for the potential to be able to expand a very good and well thought out program should we ever have a slight increase in our research funding capability. And so the idea behind the translational research was we really wanted to take ourselves, our research program, and consider the vision of let's have a future without ALS. Let's ultimately take basic research and drive all of what we're funding at least to the sentiment that we want to ultimately get to therapeutics and treat ALS and slow the disease down and someday potentially a cure, because we believe that that's ultimately what the people who are donating are wanting to get out of this. And so we are very fortunate. In 2014, as many people remember, the Ice Bucket Challenge hit. This was a phenomenon that was actually going quite around the world, or around the United States at least, for the first number of months for various other charities. But very fortunately, a number of families, including 3 gentlemen living with ALS, Anthony Sinergia, Pete Frades, and Pat Quinn, who were living on the East Coast of the United States, they really helped to steer this into an ALS initiative. And ultimately, as many people know, that actually led it to a tipping point of going viral, and was amazing because we had always hoped and believed that if the majority of people out there started to know what ALS was, they would be compelled to want to learn more and to give. And ultimately, they sure did. In fact, after the leveraging, which I'll talk about soon, and the partnerships that we formed, as you can see here, the ALS Society of Canada's research program was the second most that was raised by for ALS research, or for ALS in general around the world. And a lot of that has to do with the fact that we share a lot of similarities in our media, our culture, our celebrities with the United States. So that led to a fundraising in Canada that exceeded in a lot of other places in the world. Now, what I have to certainly mention is that when those funds came in from the Ice Bucket Challenge, they came in across the country, not only to ALS Canada, but also to our provincial partners. And we actually are in a federation with the societies that are listed here across the country. And each of those societies received the money through the Ice Bucket Challenge, and then they pool money each year from the fundraising that they have into our national research program, which was ultimately run by myself and my colleague Colleen Doyle out of our office. So collectively across the country, there was $17.2 million that was raised. And now the question was, after that, what out of those funds could be committed to research? Because there's certainly a strong amount of work that needs to be done to provide good client services and client care across the country that each of these organizations do wonderfully across the whole nation. So when we looked at those Ice Bucket Challenge funds of 17.2 million, one of the things that came up around that time in 2014 was there was some discussions with an organization called Brain Canada. And Brain Canada were the stewards of something called the Canada Brain Research Fund, which was an opportunity that the Canadian government had put forward for Brain Canada to match dollars for neurological science research. And they had proposed that up to $10 million could be matched with Brain Canada through those Ice Bucket funds. And so fortunately, the ALS societies came together after the funds were raised, when the Ice Bucket Challenge finally sort of petered out in late September, and ultimately deemed that they would deliver $11.5 million of the $17.2 million to go directly towards research. And that then allowed us to access that $10 million match and gave us $21.5 million that we could contribute to ALS research moving forward. An unprecedented amount to be sure. However, I do want to provide a little bit of important perspective before we get into what those dollars have done. And that is when we think about the $21.5 million, that is really amazing and a tremendous gift. But ALS is a very complex disease. And if we look at, I'm not trying to pick on these places in particular, but just to give perspective, the American Cancer Society, for example, in a given year can fund $145 to $150 million worth of research. Or the Towards HIV Cure Fund, for example, $268 million in 2016. And those diseases are both ones that have very viable treatment options. ALS is extremely complex, and it's going to take similar types of investments for us to be able to get there as quickly as those ones have. And if we look at just even an annual budget of the biggest funder in ALS across the world, which is the ALS Association in the United States, you know, a couple of years ago, 6 million, 3 million, or 4 million the year before that. This is about what has been going in year after year leading up to the Ice Bucket Challenge. And even in the ALS Association raising $115 million, you can see that even though this is an amazing gift, it's still something that is less than often the annual budget of other societies that have more treatable diseases. So we have to consider that when we see that impact, that it's not something that's going to cure ALS, but boy, it has certainly had a lot of impact. And one of the great things about the ALS field is that we've spent many years doing a lot with very little. So we really have been able to utilize those dollars effectively to try to get us closer to understanding the disease and getting to effective treatment. The last thing I want to focus on in terms of housekeeping before I get into what we actually funded is how do we invest those dollars? And we do it as responsibly as possible, because you understand that the donor dollars that come in are extremely precious. And we use sort of the benchmark of running competitions and having what's called peer review. And essentially this means we're getting ALS experts to come in and help us understand what are the best projects or the best individuals that we should fund with those very precious donor dollars. And you can see down here that we've actually had 3 chairs to our panel since the Ice Bucket Challenge started: Dr. Orla Hardiman, Dr. Terry Hyman-Patterson, Dr. Jonathan Glass, who are 3 internationally renowned clinician scientists on ALS that have been able to chair these panels. Now, just in very brief, how do we run these types of peer reviews? Well, it's quite a rigorous way that we do it. We take a call for applications for a particular type of competition. We give the scientists some time to devise those applications. And then after a deadline, we actually internally will determine whether or not it is eligible for the program. And we'll have a set of guidelines that will help to tell us whether or not that is actually fitting into the criteria of the competition that's being run. Then we get together a panel of experts who have the expertise to be able to review the grant applications that we received. And every one of the grant applications will have a primary and a secondary expert reviewer who will provide a written review to us as well as a score out of 7. And then after those have been tallied, we bring those individuals together so that they can sit in a room and discuss and score and ultimately rank all of the different grants that have come in or the applications for, say, a trainee award, and then have them recommend to our board of directors who they believe the best use of the donor dollars will be. So that's how we run, and that's how we deliver the donor dollars. In any other initiative where we have collaborated on something or it hasn't been through a competition, we really do go to experts to get advice on how we ultimately will deliver those dollars and what types of things we will partner on to make sure that we are stewarding those donor dollars as effectively as possible. Coming to that ALS Canada Brain Canada partnership of $20 million, it was really broken up into 3 main types of competitions, which all really were a convergence of our priorities along with Brain Canada's priorities in terms of the competitions that they run. And the first one are called discovery grants. These are high risk, high reward projects. I'll talk about those in a second. The second are career transition awards. That's that award I talked about at the very start, that we didn't know how we were actually going to fund it, but we knew that this is something we wanted to have in the field because we wanted to take a superstar young researcher and be able to help bolster them into a permanent career focused on ALS research. And the majority of the funds went to a sort of flagship program that we developed just prior to the Ice Bucket Challenge called the Arthur J. Hudson Translational Team Grant. And the idea behind this was to bring together groups of researchers to do the type of stuff that we've never been able to fund before in a collaborative effort that has the mindset of how do we start to find therapeutic targets for ALS and ultimately drive those towards clinical trial and someday hopefully treatment. The first set of those, I'm going to go over them very high level here, are the discovery grants. And the cool thing about these is that when you think about somebody who maybe not be connected to ALS, or even one of our longtime ALS researchers who has a good or novel idea, really hard for them to find someone who's willing to fund that. Now, what we can do with these discovery grants is we can fund those really cool and great ideas for a small amount, so that they can build a level of data and ultimately be able to say, look, this was a good idea, and then find other pockets of funding to be able to continue the research moving forward. So it's really sort of building that foundation of data that can help to get something off the ground. And we ended up giving 20 of these grants away. There were 4 gentlemen and 5 grants given to discovery grants in British Columbia, another researcher, Amir Sinetti Nezhad, in Calgary, in Alberta, 3 researchers in Ontario, and a number of them in Quebec, as well as one collaborative effort that came out of New Brunswick that were all funded with these discovery grants. I can't go into these in detail because then we'll be here for 3 hours, but just to give you an idea of sort of the novelty of some of these, or the cool ideas, the gentleman in the middle here, Dr. Alex Parker at the University of Montreal, he actually works with tiny microscopic worms called C. elegans, and he's made models of ALS in these worms that get paralysis and motor neuron degeneration. And actually, he was approached by a company to test probiotics. Now, not actually Activia, that's just to give an example, but certain probiotics that this company had to say, how can they learn about metabolism and obesity in their worms? Well, an interest and sort of an aspiring, a PhD student in his lab got the idea of what would happen if we put some of these probiotics on an ALS model worm. And it turned out that it actually had a positive effect. And he's actually now been able to move this forward into mouse models, and hopefully we'll be able to see whether this is something that could be a novel idea that is perpetuated into a longer term potential therapeutic strategy. That's an example of sort of the outside the box ideas. As I showed you earlier, I explained earlier, these have been funding research across the country. And just to give you two sort of more walked through examples of discovery grants that we're able to fund. The first one is Dr. Melanie Woodin at the University of Toronto that I've chosen, and a lot of these are great. This is just an example that pictorially works a little better. Now, in terms of ALS, we know that it is affecting both the upper and lower motor neurons, and the upper motor neurons are actually those that are found in the brain, and they project their long wire axons down into the spinal cord. The spinal cord then has our lower motor neurons, which are connected to those upper motor neurons, and their long wire called the axon goes out to every one of the cells of the muscles in the body to allow us to actually signal those muscles to contract or to move. And one of the things in ALS, we focused a lot on the spinal cord and on lower motor neurons over the years. We haven't really focused that much on the brain. And Dr. Woodin aimed to change that. And in fact, one of the things that we have noticed over the years, or there's been a lot of literature on, is that potentially there is an increased level of firing in these upper motor neurons that may contribute to the disease process. And Dr. Woodin had noted that there are a number of other neurons that connect to motor neurons in the brain called inhibitory motor neurons. And she wanted to know if these were impaired in their ability to sort of tell the motor neurons to slow down and stop this excessive firing. And in fact, could she be able to therapeutically target these inhibitory neurons to do their job better and actually stop or slow down that excessive firing of the motor neurons? So she has these specific compounds called DREAD compounds that she can put in the water of the ALS model mice. And this actually triggers those inhibitory motor neurons in the brain to tell the motor neurons to slow down. And in fact, she saw that there was a delayed onset of the disease in these ALS mice. It's not published yet, but it's certainly coming forward, and it's pretty interesting data. So ultimately, from this discovery grant, we found out that perhaps turning on specific brain neurons that can act as the brakes for upper motor neuron excessive firing might be a potential treatment for ALS, and is something that she's looking to potentially move forward eventually to clinical trial. Furthermore, this has gained even more momentum in recent years. Dr. Lisa Topolnik at University of Laval and Dr. Yves de Konik and another lab at University of Laval are working on other aspects to look at the same sort of concept. And actually, the PhD student who was working with Dr. Wood and Dr. Sahara Katamullah is someone that we were able to co-fund with Brain Canada through Ice Bucket Challenge Funds, as well as the Foundation Vincent Bourque and a further partnership to be able to fund her postdoctoral fellowship to work on this type of work. So again, showing that we've been able to take these discovery grants, move them forward into bigger aspects of understanding ALS, and even be able to move this forward closer and closer to potentially a human clinical trial aspect. Second one I'm going to mention is Dr. Eric Lecuyer, who is a gentleman at something called the Institut de Recherche Clinique de Montréal, which is associated with the University de Montréal. Dr. Lecuyer is a part of a big consortium called the ENCODE Consortium, which is funded in the United States by the National Institutes of Health. As a part of that big consortium, he gained the ability to have all of these tools that we could potentially use in ALS. So he was not connected to ALS before this, but he saw that these particular tools he had called antibodies, which are specific to a lot of different genes that can cause ALS, or the proteins that can cause ALS. And he thought, what if I started to use these to better understand how ALS is caused and find new treatment targets? Essentially, we use these tools in the laboratory in ALS to bind to specific proteins in cells so that we can actually look at them. Now, our immune system uses them to bind specifically to proteins that are bad, or bacteria, viruses, etc., target them to our immune system to get rid of them. We use them in the lab to ultimately allow us to visualize proteins by their specific binding. And so he collaborated with a gentleman named Dr. Jean Yeo at UC San Diego. And they're working on something called stress granules, which we're going to talk a little bit about in some future funding here and some other funding, but is really actually in the past 5 to 7 years, something that has come to the forefront of importance in understanding ALS biology. Now, when our cells are actually have stress on them, You can see here that the natural flow of things is that we have our DNA, which is our genetic information, that produces a movable form of that information called RNA, which ultimately is important for a number of functions, including producing proteins that allow our cells to do all of their normal functions and live. But when a stress happens, that RNA can be very, very vulnerable to having damage to it. So in fact, we have these things called RNA binding proteins. Now in this analogy, the RNA binding protein is the person that's doing the hugging, and it's sort of protecting the RNA. And this is actually an example from Dr. J.P. Taylor's lab. I apologize, I pressed the wrong button there. But essentially you can see that these form, and it's basically these RNA binding proteins can bind to the RNA and prevent it from getting damaged by the stress. Now these are supposed to actually break apart again, so the RNA can do its normal things and the RNA binding proteins can move around again. But in ALS, what we've learned is that there's an abnormal capacity for these to form and disassemble in the way that they're supposed to. So that's why I have the straitjacket there. So one of the things that's very important is for us to understand what is the actual components in these stress granules, so that we might be able to understand what could be trapped in there, and what also are things that we might want to target as therapeutics to make this a normal situation as in other cell types. Another way to sort of just describe what stress granules are, the way I like to describe it is, you ever seen March of the Penguins? The penguins, they all have their little functions in the colony and they're all happy and moving around, but then when a stress comes, they huddle together to weather out the storm. But then in the end, you hope that they break back apart and they can do what they normally do. However, in the case of stress in ALS, we find that some of those penguins might be really critical, but they're actually trapped in the middle of that huddle because it doesn't actually break apart once the storm has passed. And I'm not going to go into any more detail about this, but through that work, they've been able to actually come up with some pretty seminal work in very, very important scientific journals that are helping to understand what are the contents of these stress granules, how do they form and break apart, and what are the types of things in ALS that we might be able to use as potential therapeutic targets moving forward. So those discovery grants, as I mentioned, that's about 20 grants that were funded across the country at $100,000 each. That makes up about $2 million of the $20 million that we invested. Second program that I talked about were the Career Transition Awards. This was that new program we developed, and the idea really around this is, can we take a senior postdoc, which is the person who's received a PhD, is sort of working in a lab under the guise of or under the direction of a professor or an assistant professor at a university, and ultimately then wants to become their own independent lab director, assistant professor at another institution. How can we take a really promising person in that role, or a person who has just received an assistant professorship position and now has to compete and get grants with all of those established researchers out there, and help to bolster them by giving them years of funding that can keep them going long enough to build the foundation of work that they need to be competitive. And these are the 5 individuals that ultimately received those Career Transition Awards. We can say that the 2 postdocs who received them both did end up getting assistant professorship positions, so that in itself is quite a success. In the previous one that I did that took a long time, I really went through what each one of them do, but in a very high level here, the first gentleman that we've funded is Dr. Gary Armstrong from McGill University, and he's actually an expert in zebrafish models of ALS. And he's actually using a more modern technique called CRISPR-Cas to develop better and more elegant models that may better represent what's happening in the disease in these fish, and also utilizing those models to be able to better understand how neuronal firing is actually affecting the disease process. Also, Dr. Chantal Sefton was funded in 2015 through this program at University of Laval as a new assistant professor who is working specifically on a genetic cause of ALS called FUS, and particularly how that is affecting the biology of motor neurons at what's called the synapse, which is the end part of the terminal of that long axon I talked about. And this has actually been something that's been neglected a lot in our work over the many decades. A lot of work has been done at the cell body level, which is the sort of main area that's found in the spinal cord and also in the brain. But we are starting to learn more and more that it's important for us to understand these nuances that are happening at the nerve terminals. And so that's what her work is primarily focused on for her initial parts of her career. We also have 3 other individuals that were funded in 2016. Dr. Veronique Belzil, who's a Canadian, who's now an assistant professor at the Mayo Clinic in Jacksonville, Florida. She's working on something called epigenetics, which is a really hot topic in science in general, which is really understanding how the environment can affect our gene regulation and ultimately affect how diseases are started or stopped. Dr. Jihei Park at Sick Kids, University of Toronto, is working on one of the genetic causes of ALS called matron-3 and really starting to understand that better. It's something that hasn't been extensively studied yet in ALS. So through developing models that we can eventually learn more about the disease, she's been moving forward her ALS work. And finally, Dr. Kesson Patton, who's at the INRS Armand Frappier in Laval, Quebec. And he's actually also working with zebrafish models of ALS, but in this sense, more of a drug screening model to be able to see how we can eventually find drugs that can work in other models and potentially get to human clinical trial by using zebrafish as a potential way of doing very quick screening. He's also formed collaborations with an expert in developing human motor neurons from iPS-derived stem cells in California, Dr. Justin Achita, to be able to sort of form this platform for drug screening. And there's already been some successes, even though it's only been a couple of years, with some contributions to the research from each of these individuals, and many more of them have things in the pipeline that are soon to be published, and we're pretty excited about having these people as permanent contributing members to the ALS field moving forward. So that career transition award investment was another 5 individuals funded for about $1.5 million, leaving us to the Arthur J. Hudson Translational Team Grant. Now, this was a grant that was actually originally named after one of our co-founders, sort of the first ALS researcher, or the first person to really say we need an ALS research program in Canada, Dr. Arthur Hudson, along with Dr. Andy Eisen. They were sort of the 2 forefathers of the field in Canada. And it was originally intended to be a $1 million opportunity, given our normal funding pocket, to say, how can we get researchers to collaborate on bigger and better projects that are going to have ultimately more impact. But what's great about the Ice Bucket Challenge is this gave us the opportunity for the first time ever to be able to tell our researchers, dream up that project that you've always wanted to do, get together with the people that you've always thought would be helpful to bring that to fruition, and then propose it, because we have the money now to be able to fund some pretty great and amazing things. And we are able to find actually 9 Hudson grants in collaboration with Brain Canada. I'm going to very quickly at a high level walk through those 9 grants and what the impact of those grants are. The first one, and I'm only calling these Team Julien, for example, because that's the name of the PI, but these are true teams. So even though that person's responsible for the team itself, these are equal important teams that all work together on important aspects of these projects. If I highlight it that way, that's all I mean by it. So ultimately, this was a 3-year study for $2.478 million, so about $2.5 million. And this is how I explained it last time, but I'm going to do a really high-level explanation of what this is. And basically, through a number of years, Dr. Jean-Pierre Julien at University of Laval has found that a plant called ashwagandha, or Withania somnifera, which is in traditional Indian medicine, one of the active components of the root of that plant is called withaferin A, and he found that this withaferin A actually has protective qualities in some ALS mouse models and cell models. So actually, in collaboration with a company called Imstar Therapeutics, they were able to develop a compound that actually did the same thing and mimicked what was happening with this withaferin A from the ashwagandha plant. And by doing this, they then wanted to be able to test this in ALS model mice in many different ways. And then if this worked in the ALS model mice, be able to say, can we move this forward and take the steps necessary to be able to move this into human clinical trial? And ultimately, this project is then designed to, if it works in the mice properly, then be able to apply this to humans and say, can they understand whether or not there are biomarkers in the blood of humans that can tell us if it's working through the mechanism that we think it's supposed to? One of the things, if you go back to one of my clinical trials webinar, you'll see that biomarkers are a critical part of making ALS clinical trials happen now, because not only do we have to test with the drug or the therapeutic itself, we also have to be able to test whether or not it's hitting its actual target or not, to be able to know whether a false positive, or sorry, whether a positive or a negative result can be interpreted by it hitting the actual target. So in high level here, what is the impact of this grant? Well, the impact is, wow, I'm sorry about the spacing there. But the impact is ultimately that they're testing a very promising compound called IMS-088 to make sure whether it continues what Witherfern A had in terms of protective capabilities in ALS laboratory models, and then ultimately to take the steps to see whether this could be moved forward to human clinical trial. Next group is led by Dr. Sanjay Kaura at the University of Alberta. Is a huge group, as you can see, of individuals who have come together to develop through a network called CALSNIC, a project that is the largest project we've ever funded before. In collaboration with Brain Canada, almost $3 million for a 5-year study to ultimately understand human ALS better using high-powered imaging. So using MRI, and this person's actually backwards because what we're doing is they're doing a MRI of the head, but it's just to show you that they're taking MRIs of the brain to get very high-powered images of brains of people living with ALS, and doing this across the country and forming a normalized or a standardized platform of this imaging across the country. And then by taking people with ALS and without ALS, the idea is how can we start to understand why people are different with ALS from one person to another, and how can we start to understand or look at, can brain images tell us about the progression of the disease? So how can potentially a brain image over the course of one or two of these, a brain image taken at several points as the disease progresses, will we be able to see ways that can help us to say, give a better prognosis to someone about how their disease is progressing? Will there be a way that in clinical trials, this can be something that very easily tells us whether or not a therapeutic is working or not, because you could see reversals potentially or slowing of the advancement of the disease in these brain images. And hopefully someday we can learn enough from this that it might be able to be a very non-invasive way for us to be able to diagnose the disease earlier, because if certain subtle brain images can be seen or learned from this, will this be something that could be seen before someone could actually get a standardized diagnosis of ALS through the current means in a way that can allow it to happen earlier? Now, that's not what it's exactly designed for right now, but what we can learn from this can hopefully allow us to get there in the future. This is a pretty amazing project that ultimately sets up a nationwide imaging infrastructure for ALS, and hopefully to find new biomarkers that can help us to understand human ALS better, why people are different, and also how the disease is progressing. Third, we have Team Kong. This should say 2015 next to it because it was also part of 2015 investment. And this is a group of individuals from Manitoba, Alberta, and British Columbia who are using ALS as a proof of principle for a potential new therapeutic idea. So Dr. Yu-Tian Wang at University of British Columbia had developed a new potential therapeutic avenue where a specific product can go into a cell and actually target a bad protein or a toxic protein for degradation through a system called autophagy. So it basically puts a target on it and sends it to one of the cellular garbage cans. Now, if there's something that's toxic in your cells, that's a pretty good thing because you're obviously going to pull away all of that bad stuff. Now, what they wanted to do was to actually take one of the examples that we know is a toxic protein in ALS, which is SOD1 when it's mutated. In about 2% of cases of ALS, it's familial or hereditary. And then test this in cells of ALS models, and then also in ALS model mice to be able to first have proof of principle as to whether this therapeutic will actually have an effect on the disease. And if it does, they would like, that's the gentleman actually in Manitoba, as well as Dr. Yu-Tian Wang at UBC. And then Dr. Jinmin Lee in University of Alberta, and also Dr. Michael Namaka at University of Manitoba would do the initial steps of understanding how this can be moved forward into clinical trial. And there are a lot of steps when you move out of the laboratory into understanding how it works in a human before it can actually get into human testing. So ultimately, this is a new potential therapeutic method to get rid of toxic misfolded proteins using ALS and SOD1 specifically as a proof of principle. And if it works in mice, the idea was, could they start the steps towards bringing this to human clinical trial? A fourth group who were funded in 2015 is Team Korngut. This is a collaborative effort between Dr. Lawrence Korngut and Dr. Lauren Zinman, who are clinicians at University of Calgary and University of Toronto, respectively. And the idea is to run a randomized phase 2 clinical trial, double-blind clinical trial of a drug called Pimozide. And this is really an amazing piece of work by some Canadian researchers, some which I've mentioned before, working on worms, Dr. Alex Parker at University of Montreal, as well as a zebrafish researcher at University of Montreal as well, Dr. Pierre Drapeau, who was the supervisor to the two gentlemen I talked about before who mentored those individuals, as well as Dr. Richard Robitaille at University of Montreal as well, in mice. And in all three of these systems, something called Pimozide was seen to have a potential protective effect on ALS. And so the idea was, in collaboration, they came to Dr. Lawrence Korngut and said, could you put this into clinical trials to potentially see if it can slow progression of ALS? And now the reason why this could move straight into a phase 2 clinical trial from drug screening in a lab is because this is a long used schizophrenia drug called, well, Pimozide. And so it's actually gone into clinical trial at 100 participants. It's currently still recruiting at 9 sites across Canada. And we eagerly await finding out whether or not the Pimozide will have a significant effect on slowing progression of ALS. So ultimately, with this particular grant, it's testing whether or not Pimozide can slow progression of the disease. And it's also, as we've talked about a lot before, it's also testing whether or not a potential novel biomarker, which can help to explain whether or not there's a strengthening between the motor neurons and the muscle at what's called the neuromuscular junction, could potentially be something that not only could help in this clinical trial to see if the Pimoside's working, but also be something that might monitor progression of the disease and other potential neuromuscular junction strengthening drugs in the future. Another team that we funded in 2015 is Team St. George Hislop, led by Dr. Peter St. George Hislop at the University of Toronto, and collaborating with another individual here in Toronto, as well as someone at the University of Laval. And the idea here is to actually target those stress granules I talked about, but in a very unique way of something that I'll explain in a moment, through a $1.5 million 5-year study. Now, something that we've only really known about in biology, let alone in ALS, in the past several years, is that a lot of the proteins and RNA in our bodies or in our cells actually aren't always solid. They actually can go into a liquid tight state before they become solid. And so I showed you this before, where when there's a stress upon the cells, the RNA binding proteins actually can hug the RNA to sort of protect it from any kind of damage during that stress, and then they're supposed to relieve and break apart. So this is an example of that happening. However, what you can see there is these do kind of look like liquid droplets, and it turns out that they actually are. And so there's a phenomenon called liquid-liquid phase separation, which wasn't really known in biology until the recent, even less than a decade ago. And it's almost like an oil and water reaction or a lava lamp, where the RNA and the RNA binding proteins form these sort of liquid droplets that can then dissociate or disassemble back into the normal situation after the stress is gone. However, as I said before when I was speaking about Dr. Lecuyer's work, when the RNA binding proteins hug the RNA in ALS, they tend to actually get stuck there. And in fact, what we have is a situation where they actually gelatinize from a liquid into a more gelatinous state, which can trap many important proteins within, or proteins and RNA within them, and might be potentially toxic for 2 reasons. One is it could potentially disrupt what's happening in the cell. And the second thing is it may be trapping things that are important for the cell and the motor neuron to actually survive. And through this work, Dr. St. George Hislop has already published a very important study on our understanding of how these stress granules and how this phase separation affects ALS, including another one that is not yet published but is soon to come out, which is in another top-tier journal, a very important, impactful work. And he's also developed, found some interesting targets in that might be able to be moved forward, not only in just understanding RNA biology in ALS, but potentially being therapeutic targets as well. And finally, in 2015, Dr. Christine Van de Velde has led a team to also work on those stress granules, but from a different standpoint. So we've really learned, as you're clearly probably gleaning from this, is that stress granules and RNA biology are very important to ALS pathogenesis. And in fact, this also comes from the fact that what we've learned is many of the genetic causes of hereditary ALS are due to mutations in RNA binding proteins. That's why a lot of the work by Dr. St. George Hislop's team, Dr. Lecuyer, and now Dr. Christine Van de Velde's team are all really important for us to understand the disease and to be able to therapeutically target it. This team, in fact, this is how I showed it before, a little too comprehensive for this webinar in the time that we have. They're really focused on how stress granules form and dissociate, particularly around a protein called TDP-43. Now, I want to show you quickly why TDP-43 is so important in ALS. Now, this is a protein that in a normal motor neuron is found inside the nucleus where our DNA is, and it's sort of the command center for the cell. And it has its functions really largely within the nucleus, as well as a little bit outside of the nucleus, but it shuttles back into the nucleus normally. However, in ALS, what we see is that the majority of the TDP-43 actually can move outside of the nucleus in certain cells that might be susceptible. And this can actually clump up, as we've seen with those stress granules, and ultimately form these sort of structures that trap important proteins and RNA within them, and also might be disrupting the functioning out here in the cytoplasm. But what's really important about understanding TDP-43 biology is that it is actually abnormal in 97 to 98% of cases of all ALS. In certain motor neurons in those individuals, in most individuals with ALS, there is some form of TDP-43 pathology. So understanding what it's doing is extremely critical, very likely critical to us understanding the disease better. And Dr. Van de Velde had been working with stress granules already and a particular protein called G3BP1 prior to this funding. And while this work isn't published yet, what I can say is I have seen at a recent conference the culmination of the work to date from this Hudson grant, and is pretty impressive in terms of what is revealed in terms of understanding ALS stress granules with regard to TDP-43, and also a potential therapeutic target that might someday be able to be moved forward or move into some form of therapeutic targeting for the disease. So ultimately, it's designed to help get a more comprehensive understanding of TDP-43 stress granule biology, as well as identifying a potential therapeutic target to be able to normalize that stress granule activity in biology, which, as we've now outlined, is abnormal in motor neurons for people living with ALS. So in 2016, we ran the program again. Dr. Janice Robertson led a team that was funded to study a particular genetic cause of ALS called C9orf72. This was discovered in 2011 as the most prominent genetic cause of ALS, and has become a very big part of our ongoing understanding of how the disease is caused. And this is how I explained it in the past, but I'm going to really simplify this for you. Now, what we have learned over the years at a high level for C9orf72 mutations, when somebody has these mutations in ALS, and that makes up, as I said, the most prominent genetic cause of the disease, when they have this mutation, there are two things that may be happening. One is it's actually reducing the amount of C9orf72 protein that is produced. So it could be causing a loss of the ability of its normal function to help motor neurons survive. Unfortunately, when we discovered this mutation in 2011, we didn't know what the normal function of the protein is, so we still have to determine what that is. And also, the mutations cause additional toxic functions. So both by producing some toxic RNA and toxic proteins, this can also affect motor neurons in a way that's not healthy for them and can lead to ALS. So what we are starting to believe is it's very possible that it's a combination of all of these things. And there are a number of people in other countries who are working a lot on the toxic function part, but there hasn't been as much work that's been done on understanding the loss of the normal function of the protein. Dr. Robertson's team is actually working very hard on comprehensively understanding what is that normal function, and then ultimately what can that be doing to motor neuron biology that might be conferring ALS. The idea is if the mutations are preventing some of the normal functions of the protein, can we then reinstate these as a potential treatment target in the future? Next, in 2016, we also funded Dr. Guy Rouleau to lead a team. He's out of the Montreal Neurological Institute at McGill University, as well as 2 other institutions where he has collaborators in Quebec. And a 5-year study really focused on understanding 2 different cell types, how they intercommunicate, and a specific type of cell in cultures in the laboratory. So this is how I explained it in the past, but really the high level talk on this is around a combination of 2 cell types, motor neurons, and also another cell called astrocytes. So one of the things we know in ALS over the years is that ALS is actually not just a disease of motor neurons, it's the degeneration of motor neurons that actually leads to the symptoms of the muscle pathology, the degeneration of the motor neurons leads to the symptoms of ALS, including the loss of the ability to move the muscles, eventually the paralysis and the terminal aspect of the disease. The other cell types around it, including astrocytes, are extremely important to proper motor neuron health. In our normal bodies, the motor neurons and the astrocytes have a crosstalk where the astrocytes are sort of like their personal assistants. And so actually using a technique that has been developed over the past decade in science and is moving forward to become likely a very critical aspect of understanding a lot of diseases is the ability to do what are called iPS-derived cell types. In this case, iPS-derived motor neurons and astrocytes. So what you can do ultimately is take either skin or blood from an individual who lives with ALS or without ALS, and you can turn these cells into stem cells, and then ultimately turn them into whatever cell you want to study in the body. And in this case, Dr. Rouleau's team is going to turn these cells into human motor neurons and human astrocytes that have the same genetic makeup of the individual who provided them. And using multiple different types of ALS backgrounds, so different genetic forms of ALS, sporadic ALS, all of this, they'll produce cultures of both the astrocytes and motor neurons to be able to understand what is the crosstalk between them, something that we've not been able to do effectively using human cells to date. And also, once this is understood, how can this be used as a potential drug screening platform to normalize this or to affect this in human bodies eventually in clinical trial? So ultimately, the impact here is that understanding how human motor neurons and astrocytes communicate, as well as using these cultures to potentially screen for treatment targets in the future. The last one that I'm going to talk about is Team Durham. In 2017, this is led by Dr. Heather Durham in the Montreal Neurological Institute with 3 other collaborators. And at a very high level, not this slide, essentially the work that Dr. Durham's been doing for many years focuses again on that idea of how does the cells respond to stress, but in this case, not necessarily with RNA biology, but in this case, protein biology. And when a stress hits one of our cells, it can actually cause proteins to lose their normal function. So, or to actually gain a toxic function. So proteins in our cells do most of the major activities that keep us alive. And they have this normal 3-dimensional structure that allows them to have the normal activity. However, when stress has hit them, they can actually misfold or unfold, and that can actually cause them to either lose their normal function or gain some toxic function. And our cells actually protect ourselves against this because we produce things called heat shock proteins that come in and refold the proteins so they can continue their normal activity after the stress is gone. However, Dr. Durham's career has really focused on understanding that motor neurons are very much impaired in creating these heat shock proteins in the way other cells are, and this might make them selectively vulnerable. And over the years, she's actually been able to understand how to actually restart that system through a combination of drugs, and using her collaborators in this project, she's going to do multiple testing of this combination of therapeutics to be able to see whether or not the heat shock protein response can be reinstated in these motor neurons, and then ultimately, can that be a protective thing that can be moved forward to clinical trial? So again, can a combination of particular drug types come together to elicit that protective response? And then if it can work in ALS laboratory models, can that be something that eventually gets moved forward to human clinical trials? So a pretty successful Hudson grant program with Brain Canada that funded across the country, but it becomes even more apparent how important this was And you see that across the teams, there are 44 different researchers in 5 different provinces that were able to be funded by the Hudson grants. Moving forward, if we add that funding up, we can see that we're almost up to the 20 million. Pretty amazing funding and impact that comes out of that. But wait, there's more, as if you've been doing your quick math calculations, we still have 207,786 left. But what I can say is that in a few of those instances, there has been situations where we've been able to further leverage funding through other partnerships, be able to get a little bit more funding back and actually be able to run one more program in 2018 with Brain Canada that allowed us to fund some trainees. And what do I mean by this? Well, that means PhD students who are very promising and working on ALS projects in prominent ALS labs for 3 years of salary funding, and we're actually able to fund 3 of them. I would love to be able to talk about each of these, but we actually have an ALS Canada blog on our website, which I encourage you to go to, because those will do a much better job of explaining each of these projects and the individuals behind them than I would do in this webinar. And secondly, our postdoctoral fellowships that we co-ran in this program with Brain Canada, and that was allowing us to fund 2 individuals, Dr. Sahar Kadamala and Dr. Yulong Sun, who again also have great blog posts available for you to be able to read on our website. Now, after that 20 million, you're saying, well, what about the other 1.5 million? Well, don't worry, we're getting to that as well, because we are pretty excited with some of the investments that have come from those funding as well. One of the first things we did, we ended up funding actually 4 what are called bridge grants. And this was because at the current time, when the Ice Bucket Challenge hit, there were a lot of changes happening in the government funding system. And there were a number of key ALS projects and researchers that were not able to get funding and needed more momentum to be able to continue working on these promising projects. So we were able to fund 4 of these bridge grants to the individuals that are listed here: Dr. Neil Cashman, Dr. Avi Chakrabarty, Dr. Yasna Kriz, and Dr. Victor Rafuse. But what the heck is a bridge grant? Well, ultimately, as I've just said, when the government funding system called CIHR funds research, it turns out that a lot of great research just isn't able to be funded by the government. And so some of those researchers have great work and are ALS researchers, but don't actually get the funds to continue their work. And these bridge grants allow them a period of time to develop more data to ultimately try to get into the top tier of funding. Just to give you an idea of sort of the success rates here from the government funding system, here's an example from last year. I think this is actually the most recent competition in spring. There were 2,445 applications and only 382 were able to be funded. So you can imagine how many great projects are left on the table. And these bridge grants help to provide bridging funding to reapply in subsequent years. That's only a 15% success rate. One of those actually has led to something pretty remarkable. So over the career of Dr. Yasna Kriz at the University of Laval, she's been working with the immune cells of the brain, particularly cells called microglia. And she's been able to learn a very nuanced way in which we might be able to turn those cells in ALS back to the healthy way that they are in a normally functioning central nervous system. And by targeting something called SRSF3, she actually has now some investment from a company to be able to potentially move this forward as a therapeutic target into clinical trials. So that's pretty exciting stuff that has come out of already just after a couple of years, some of the work that's been funded through Ice Bucket funds. Furthermore, we have become a part of a huge international consortium, one of the biggest undertakings in ALS research anywhere in the world in history, which is called Project Mine. This is an unprecedented effort to take 22,500 what are called whole genome sequences to be able to fully understand the genetics of ALS, even the genetics of sporadic ALS, which I don't have time to explain right now, but will be extremely critical to our understanding of the disease moving forward. And actually, we are able to fund $250,000 towards that effort, and Brain Canada actually chipped in an additional $150,000 matching to be able to help move forward on Canada's drive to our goal of delivering 1,000 whole genome sequences to the grand total in the end. And this is really just an example of how we can share resources and leverage financial capabilities around the world, with 20 countries being involved in this program, be able to do something that's truly going to have impact on ALS. And ultimately, to this point, they've already published some new genetic causes of the disease. And as we get closer and closer to the 100%, they're going to understand more nuanced things about the genetics of the disease in the future than we've even been able to have the capabilities to do in the past. Another thing that we've been able to fund, which is tremendous, which is a great collaborative effort and is a very busy slide, is something called the ALS Reproducible Antibody Platform at a very high level. What's great about this is not only is it going to provide a tool or a set of tools for the entire global community to save money and be more efficient on research, it's also a great new collaboration between ourselves, the M&D Association in the United Kingdom, and the ALS Association in the United States. And together, through funding this ALS RAP, or Reproducible Antibody Platform, they're creating a system where we can have bona fide tools to use for different targets in ALS, so that people aren't buying commercial tools or antibodies for their lab and finding out later that the one that they bought didn't work very well and wasting a whole bunch of time and effort and money on those. This is actually designed to massively increase the value of donor dollars in the future, and we think is a pretty exciting thing moving forward. Finally, the best practice recommendations is something that is coming forward soon. This has been a monumental effort by clinicians across the country, led and chaired by Dr. Kristen Shoesmith out of the London Health Sciences Centre, to develop what the United States and what Europe has already done, which is a set of guidelines or recommendations to say this is the standard of care which a person for ALS should have. So multidisciplinary care, treatment ideas, all of this put together into a document that can be used not only in Canada for leveraging hospitals for meeting those standards of care, but also advocating in provinces for better care for people living with ALS around the country. And so this is ultimately a research project because it is a huge effort into going to the literature and combining the expertise of these neurologists with the data that's been discovered through research over the years on the best care practices, putting these together into a comprehensive document that can ultimately help Canadians across the country in getting better or closer to an ultimate standard of care. So stay tuned for that, because there's gonna be a lot more on that coming in later this year and early next year. We're getting close to the end. I know I'm at 12:57. A few final metrics here. Before 2014, the most number of applications we had in one competition was 9. I don't know why I had that image there. Now you can look over the course of the total Ice Bucket Challenge funding that we're able to put forth. 310 total applications came into this. This tells us that not only were new researchers coming into the field of ALS as a result of the Ice Bucket Challenge with their ideas, but also those longtime ALS researchers were able to apply with things that they never thought they would be able to do in the past because of the lack of funding to be able to do it. Furthermore, 123 unique researchers applied to programs across the country, and actually some semblance of funding went to 66 unique researchers, which is equivalent to 54% of the researchers who applied got some funding through Ice Bucket Challenge. I would say that's a pretty great impact. Total grants funded, 43 total grants, including multiple international collaborations, and something I'm going to tell you about in a little bit, because I think it's not just limited to the 43 that are funded directly through Ice Bucket Challenge funds, but something that we're able to do as a result of those funds, which is to leverage the ability of our other funds to go towards even more programs. But wait, there's even more. We may need to stay tuned, because even some of the funds that we're able to further leverage from those 43 grants in total means that we may be able to announce a couple more things using that partnership with Brain Canada in the near future, and we're hopeful to do that very soon. So I don't have a lot of time. Typically, I like to go through these one by one, but just to show you that the impact so far has actually been tremendous. And if you were to speak to a researcher even in 2013 as to where we are versus where we are now, it is a monumental change in our understanding of the disease and our potential to have treatments for it in the foreseeable future. And a lot of that has come from Ice Bucket Challenge funding. And we have so much more to gain in the future. We have therapeutic targets that are directly funded that will be coming out of Ice Bucket Challenge funding. We have an actual clinical trial that's going to have results coming in the near future. There are 5 new laboratories that are working on ALS moving forward as a result of the Career Transition Awards. Great new partnership with Brain Canada that we can hopefully move forward into more funding in the future. Our connection to international efforts like the ALS RAP and Project Mine. The best practice recommendations are going to be amazing when they come out in terms of the impact that they can have across the country. So there's so many things that have had already impact on the field of ALS, and I think it will only continue for the next several years to have more and more impact. And this just says, if we look at the perspective of what people were saying back in 2014 about where my dollars have the greatest influence, I would argue now that they've had a lot of influence, and actually more influence than they could have had in other areas. So we have to remember that all of this came from the equivalent of about 6 months investment for other treatable disease health charities. So that is an awful lot of strong value that has come out of the wonderful gift that the Ice Bucket Challenge is. And if anyone has questions about specific aspects of these, I'd be happy to talk about them offline. I know it's 1 o'clock, so if you have to go, hopefully you got the gist of it. I've got 1 or 2 quick slides here left. One of the key pieces is, if you even donated $1, or even if you did the Ice Bucket Challenge or are aware of it and spread awareness of ALS, you should be proud and take ownership of not just what I've told you now, but also the fact that that IBC, the Ice Bucket Challenge, allowed us, as I just said, to leverage the money from our walks for ALS across the country into doing even more than we could have done before. So we were able to put forth a clinical management grant to say, how can we use research to do studies that will allow us to better care for people living with ALS in Canada and around the world. That would never have happened without Ice Bucket Challenge. Clinical research fellowships to bolster our ability to have better clinical researchers and clinicians, neurologists across the country. And also even more training awards and bridge grants that weren't announced here, that were not funded directly through Ice Bucket Challenge, but certainly indirectly. And the advancements, and there have been many since 2014, should ultimately be owned by anyone who gave, because this has all come together to make a huge change on where we are in ALS research in 2019. Last slide, the future is something called Capture ALS. This is just a teaser. Where do we come from all that Ice Bucket Challenge funding? You know, we've now come back to our annual levels that we have from our donor dollars, but this has allowed us the capacity and the resource development over the years, as well as the connections and the collaborations to build something that's truly national and can have an international impact. And something called Capture ALS, which stands for Comprehensive Analysis Platform, Understand, Remedy, and Eliminate ALS, is a platform that's going to help us to understand human ALS heterogeneity. Why do some people have slow disease? Why do some people have fast disease? Why do some people have cognitive impairment while others do not? How do we understand that? Because that is one of the last remaining major hurdles that's going to allow us more efficient translation of these great therapeutic targets from the laboratory into effective clinical trials. And the idea is that Capture ALS will help us to gather in Canada the most detailed biosignature of a human with ALS ever. I would like you to please stay tuned to that. We're going to probably launch that in 4 sites moving forward. The idea is that it will be open science sharing of the data. So it will be something that de-identified data can be shared and mined around the world so that people can advance this as quickly as possible, and using modern machine learning techniques to something that didn't exist as it is today 5 years ago to the same capacity. How do we put that forward to understanding these very complex biosignatures of why human ALS is so unique and so complex? And ultimately, stay tuned for more on that. I'm just excited to be able to tell you that, you know, a lot of the Ice Bucket Challenge stuff has led to the capacity to be able to do a program like this. And that's it. That's all I have. I apologize for going 3 minutes over time. That's as quickly as I can go through it all. There was a lot of funding there. I am happy to take any questions that anyone has. I'm getting delivered some questions now. Please, I will stick around as long as it takes to be able to answer any of your questions. Okay, so the first one is, what field are the experts experienced in? Medical research, finance? I think you're referring to peer review panel. Oh, sorry, that note was the person is probably referring to the peer review panel. That is a great question. And one I didn't delineate. Yes, these are ALS experts. So the idea is we really want to bring together people who have diverse expertise in the field of ALS to be able to make sure that we're getting people who understand where the cutting edges of the field. What are the things that are most going to have impact on our global understanding of the disease and our ability to treat it? So we'll have people who are more pure clinicians with ALS because they're going to help us to understand that aspect. We have a number of people who are both clinicians and scientists, so they will have a lab as well as they see and treat people living with the disease. And we'll also have a number of people who are working on the laboratory or basic side of research to have a very broad panel that can discuss and hopefully put the money towards or recommend the money going towards the best possible ALS research. We have thought about expanding the panels a little bit, but what we really want to do is make sure that we are, funding the stuff that's going to have the most impact on a global scale. Happy to talk about that more offline if whoever asked that question wants to contact me. I should also mention dtaylor@als.ca or research@als.ca for any additional questions that you have, or you can still fill them into the webinar. We are still monitoring it. Okay, why spread the money so thin in so many directions rather than bringing to Canada promising treatments like Neurone, Treg, or copper ATSM? That is also a great question. So the first part of that question is we still don't understand ALS fully. And so if we don't fund the preclinical research, we are not going to ever get to an actual treatment that we know really works. On top of that, though, the three that are mentioned there, I do talk about them extensively in the clinical trials webinar. First of all, there was no asking for the funding of those particular ones. A couple of them didn't really exist at the time of the funding of the ALS, of our funding of the research through the Ice Bucket Challenge. And furthermore, in the case of funding a particular therapeutic, we did fund Pimozide, that is true. We had an application from Pimozide, and the peer review panel did decide to fund this. This was an academic clinical trial. So had we been proposed to fund, for example, T regulatory cells for an academic clinical trial, that would have been something that would have been considered if it had been through a Canadian researcher. But ultimately, when it comes to understanding the disease, this is where we can have the most impact with the dollars that we have. If we put all of the funds into a particular segment of a therapeutic, and then that therapeutic doesn't end up working, then we have just used all of the donor dollars that have been given to us through one particular aspect that we don't know works. And I think that's the key piece, and maybe that was a bit of a muddled answer. I'm happy to talk about this with whoever asked the question offline. We can have a call. But in the end, I think with each one of these, we're excited about them. With Neurone, it's at phase 3 clinical trial. We're hopeful that it works, but there is no proof that it actually works. There's no scientific statistical proof at this moment. And so we're hopeful that it works, but if to put all the money into something that we don't know works would also be very difficult if it turns out that it doesn't. Secondly, the T regulatory cells, again, is very exciting, as well as copper ATSM, but those are so early in the development. Again, there's no statistical evidence yet that they actually work. And we've seen this time and time again in ALS, where there have been things that are promising and they've ultimately led to a failure. Now, I hope all three of these work really well, and I can totally understand why someone would want the money to go there. But in the end, we have to do by best by what we feel and what the field feels is going to get us to treatments that are effective and eventually a cure for ALS as fast as possible. And that involves putting things into understanding the disease effectively while knowing that something like Neurone is going on and the phase 3 clinical trial is happening, and we are excited to learn what will ultimately happen. So In the end, if there are further questions that or anyone wants to discuss that offline, let's have a conversation about that. And I please urge you to go to my clinical trials webinar that I gave about a month ago, as that really does talk about each one of these in more detail. I'm very excited to see what will happen with all three of them. Were any of the funds from the Ice Bucket Challenge used towards equipment loans, or did it all go to research? So as I mentioned at the start, that $17.2 million was ultimately raised across the country. And through our wonderful federated partners, the other ALS societies across the country, the other provincial societies, $11.5 million was committed to research to access the match with Brain Canada. The remainder of those funds did stay in the provinces, including ALS Canada for Ontario and the other provinces, to be able to advance the ability to care for people living with ALS in their provinces. And some of that stuff has gone to, programs such as equipment programs. Some of that has gone to direct care models, all kinds of wonderful things that have been put forward to hopefully advance our ability to a better care for people living with ALS today, because certainly with research, we haven't been able to help a person who is living with the disease at this exact moment. So there is definitely a balance in where the funds has gone overall. Fourth, where will be the main source of funding in the future? Where will it come from? That's also a great question. Well, one of the places that the majority of the funds will come from is our national Walk to End ALS. I apologize if I said Walk for ALS earlier. It is the Walk to End ALS. Please go out and support those, as well as other opportunities to fund research. So we're looking to major donors. Can we find people who are looking to fund large initiatives or specific projects, or even to fund research in general across the country, and other avenues in which we can fundraise specifically for research or research projects. But yes, ultimately, most of the research funding will come back to where we were at before, but we are endeavoring forward to try to raise more funds for research all the time. I certainly believe that, and I've said it many times, this is a disease that will be treatable in an effective manner. It is a matter of when, not if, that it will happen, and that when really does have a huge dependence on the ability to fund it. It is a very complex disease, extremely complex. And medicine has really figured out a lot of the low-hanging fruit. Neurological diseases are difficult, but we are getting closer to understanding them, and we are going to reach a time of treatment. The more funding that we can get towards getting there is just ultimately going to get us there faster. Okay, moving forward, we have a fifth question. Are there certain sources of funding, example government, that should be contributing to ALS research that isn't? Well, okay, so I'm clearly very biased, and I think that it would be great if everyone contributed to ALS research, because it's such an underserviced research, a complex disease that needs more funding. But yes, there are certainly sources that could be contributing more to ALS research. We have certainly put forth a proposal to the government to help us fund Capture ALS. We do go and do a lot of federal advocacy to try to get the government to put more funds into ALS research, and that will continue. We will always be doing that work to try to move that forward. We also are continually wanting to find partnerships. Brain Canada being a fantastic partnership for us that has actually used some government funds, has used government funds to help leverage and fund ALS research. But other organizations, the Foundation Vincent Bourque I mentioned before, and there are several others that we're looking into, the ALS Association, the MND Association. How can we take other ALS societies around the world? And our CEO is part of the board of directors for the International Alliance of ALS Societies. I'm also the chair of the Scientific Advisory Council for them. How can we start to think about more global collaboration that's going to bring Canadians into or plugged into what the world is doing, so that we can leverage each other in a way that can have more effect and impact. I think we are moving towards that in certain ways, and I think there will be more sources of funding that will come to the forefront. But the Ice Bucket Challenge was just an amazing one-off gift, and anyone that does them now, we're still so grateful, because it certainly brings awareness and some funds towards ALS research. So hopefully that helped to answer your question, but I'm certainly, again, in all cases, happy to talk about it more offline. Okay, 6th question. Is research done on patients who had physical trauma before their diagnosis? Okay. Specifically through the Ice Bucket Challenge, we don't have anything that's directed upon any kind of trauma-related ALS research, but these things do exist. Basically, there are a number of ALS researchers around the world who have anecdotally seen that people who are in high-impact sports, people who are in the military, people who have had anecdotally head trauma or other body trauma have developed ALS. And so there's a consideration that perhaps there's some link for some people who are genetically predetermined to get ALS, that trauma may actually trigger the disease or trigger some disease process. In fact, that's why it's so important that Project Mine goes forward and something like that, because eventually we may be able to understand what is the genetic key cut of an individual that makes them susceptible to trauma, for example. Anyways, these studies have been happening, and to date it's inconclusive that we have a specific answer as to whether there's a relationship, but it is such a strong anecdotal evidence that these works are still continuing to move forward. And I think another piece is when we move forward and think about that last big hurdle, or one of the big hurdles that remains in understanding ALS, which is human ALS. Why is one person so different from the other? This can be certainly an important aspect of a biosignature, which is what are the environmental things you've been exposed to? Have you encountered some sort of physical trauma in your life? And then how can we weave that into that signature that tells us whether or not physical trauma or head trauma is connected to developing ALS for some people? So while there isn't a direct funding of that, there is definitely a drive forward in the field to better understand that. And as a result of the Ice Bucket Challenge, things like Capture ALS will hopefully have some relationship to that, if not a direct one. So that is the end of the questions that I have right now. As I've said, once again, if you have questions that come up over the course of the day or in the next while, please feel free to reach out at research@als.ca or dtaylor@als.ca. We can set up a phone call, we can have a chat if you have any questions or would like further information on the questions that I've answered. And so I'm well over time. Thank you for anyone that's still out there with me, and I look forward to doing more webinars in the future and interacting with all of you. Thanks, have a great day.